Unit says
Genetics and Cancer
by Rachel
and Adia.
There. My name is Rachel, and today we're going to be talking about meiosis. You're probably wondering what meiosis is. Huh? Well, I'm going to be talking about meiosis right after this quick break.
The purpose of meiosis is to preserve the diploid number of chromosomes in an organism. Each parent from that organism must contribute only half of its chromosome, and to do that, both the female and male must produce a gamete to create a zygote. First, the diploid cell would go through interphase, which creates a double-stranded chromosome. Then it would go through meiosis, which involves 2 rounds of cell division, meiosis 1 and meiosis 2. When entering meiosis 1 and 2, there are 4 stages. They are called prophase, metaphase, anaphase, and telophase. In prophase 1, the chromosomes line up side by side, and that creates homologous pairs. This process is called synapsis. After synapsis, the chromosome pieces are exchanged between the homologous pairs, and that's called crossing over. Come on. Okay. So, this provides genetic variation, and by that, the chromosomes will have several alleles or different forms of the same gene, which is called recombination. In metaphase 1, the chromosome pairs are now lined up at the metaphase plate. And their alignment is random, which means that the copy of each chromosome that ends up in the daughter cell is random. So how does this process connect with cystic fibrosis? Genes on these chromosomes code for specific proteins that control body function. There is a gene on chromosome 7 that is responsible for the production of protein called cystic fibrosis transmembrane regulator. Unfortunately, in cystic fibrosis, a gene mutation or a non-disjunction has occurred on chromosome 7. This can lead to an absence or defect in the CFTR production. Cystic fibrosis is a recessive gene, so that means that CFTR gene must both be mutated. Having a heterozygous gene for the disease means that you are a CF carrier. Carriers do not have the symptoms of disease, but they can pass it off to their offspring. Both parents must be carriers or have the disease in order for their child to have the disease. The pedigree shows the likelihood of having cystic fibrosis. This karyotype is on chromosome 7. In respiratory symptoms, some symptoms can be persistent coughing, and in digestive symptoms, some can be poor weight gain, and some of the treatments like medication would be antibiotics and chest physical therapy, pulmonary rehabilitation, which can be exercise training and surgery.
Hi, I didn't see you there. My name's Adia, and I'm gonna be showing you guys the ways of mitosis. Vámonos, muchachos.
The purposes of mitosis include producing daughter cells that are identical copies of the parent cells and to maintain the proper number or organisms throughout the generations that go by. Interphase is the first stage of mitosis, and it includes G1, S, and G2 phase. G1 phase creates proteins needed for DNA replication. S phase is where DNA is actually replicated, and G2 phase is where microtubules are actually produced. Prophase is where the spindle fibers from each centriole attach to each sister chromatid at the kinetochore. Metaphase is the stage where chromosomes line up in the middle of the cell. Anaphase is when the daughter cells are created with identical sets of chromosomes. The last stage is telophase, in which mitosis reaches its goal in making 2 daughter cells. Checkpoints are used to control whether a cell is ready to go on to the next step of the cycle. The checkpoints include G1, G2, and metaphase. The cell cycle clock controls the pace of the cells during each cycle. Kinase is the protein that gives the green light for cells to go on to the next phase in the G1 and G2 checkpoint. External cell division, like PDGF, is used when an injury occurs and platelets release PDGF, which bind to fibroblasts in order to process the healing of the wound. Unfortunately, cancer cells are unable to respond to such control mechanisms. Cancer cells lack contact inhibition, which causes them to keep growing for no reason, basically. They start invading other tissues and keep growing when they don't need to. Benign tumors don't spread and are removed very easily and are non-cancerous. Malignant spread everywhere and are hard to get rid of and are cancerous. Ewing sarcoma is a malignant tumor found mainly in the bones, soft tissues, and muscles. It occurs mainly in teenagers, and the symptoms include pain and swelling. On average, there's 250 cases of Ewing sarcoma diagnosed in the U.S. each year. Chromosome translocation is an abnormality created due to the wrong rearrangements of non-homologous pairs. In this case, chromosome 11, EWSR1, and 22, FLI1, created an abnormality in the translocation process on a specific gene, causing the malignant tumors to form.
You there. What was my line?